Okay, well, thank you very much for the invitation to speak here. I am Karina's brother, as you probably noticed, and I was also Kevin Foster's postdoctoral investigator, and now I'm at Sloan Kettering for 6 years now. And I've been focusing on this problem of ecological dynamics in the gut microbiota, and especially using as a model system bone marrow transplant patients from Memorial Sloan Kettering. We don't do just that in my lab. We actually have a hybrid computational experimental lab, and we focus primarily on three problems. The first one is trying to understand how bacterial cells come together and do things collectively, such as building biofilms or swarming. We also look at very similar problems, but in cancer biology. So how do cells within a tumor come together and engage in collective invasive phenotypes, and especially how do they interact and many times co-opt normal cells of the immune system that actually help the cancer proliferate even more. Now, the problem that I'm going to talk about today is a problem about the gut microbiome, and particularly colonization resistance. And just to start and continuing on the theme that Marvin left off, I'm going to bring up some examples from the 19th century, and this time of microbe hunting, where basically we were trying to find microbial causes of disease. Ultimately, the goal is to try to understand why some people get sick while others don't. How do certain sicknesses pass on from person to person? And I learned a lot about that from this book that Kevin Foster actually gave me before I left to Sloan Kettering, called Microbe Hunters. It's a beautiful book. I imagine most of you might have read this book. And it tells the tales of Van Leeuwenhoek and Pasteur, but also Koch, which I'll bring up today, and the fact that Koch was very much focused on constructing a set of very well-defined postulates by which you can determine the microbial cause of a disease. And I believe this type of approach, the Koch postulates, were a really important framework that enabled a lot of the molecular biology, and especially determining causes of pathogenesis throughout the 20th century. But at the same time, it left several questions unanswered. And so some of those questions are what we want to address today. One of Koch's rivals at the late 19th century was Max Joseph Pettenkofer. And one of the questions that he posed to Koch was he asked Koch to send him a bottle with Vibrio cholera, because he didn't believe that Vibrio cholera would be the causative agent for the disease, and he actually drank it. And according to this story, he actually didn't get cholera. And so the question is here, why didn't he get cholera? So there's possibly several explanations for this. One of the explanations is that he apparently had a previous bout of cholera, and so probably developed some level of immunity. I read a bit about this in the internet, and I was also find very interesting ideas that perhaps he drank a very high-density solution of Vibrio cholera, and so this quorum sensing might have been turning off the virulence of Vibrio cholera, which is an interesting idea. But possibly another explanation is that his gut microbiota resisted colonization by Vibrio cholera, and maybe the quorum sensing is involved in all of that. But basically, what I want to address here is how does the gut microbiome confer resistance to colonization to certain pathogens. And when we look at the variation that all of us have in our gut microbiome, can we understand something about how that determines whether each of us gets sick or not? Even though we get exposed to the same microbes, some of us will get sick while others don't. The principle behind colonization resistance is actually quite simple. It's an ecological idea. Your gut is colonized by a very diverse microbial ecosystem, and this ecosystem is somehow, through some features that we don't fully understand yet, a resistance to invasion by a foreign species, pretty much like natural ecosystems will be resistance to invasion by a foreign species. So we have here an example that I took from a chapter that I actually wrote for this book, Virulence Mechanisms of Bacterial Pathogens by the American Society for Microbiology, that is coming out now during the ASM General Meeting in Boston. And the chapter that I wrote about was on social microbiology and pathogenic bacteria, really to try to stimulate microbiologists in general to think about this type of ecological interactions and evolutionary principles that occur in microbial populations. But so the principle of colonization resistance is very simple. You have a gut that's colonized by basically an ecosystem of microbes, and somehow that resists invasion by pathogens, for example, Clostridium difficile represented here. Now, what we're also very concerned in a hospital setting is what happens when the patients take certain antibiotics and the effect that these antibiotics have in the stability of this ecosystem. They can end up disrupting a lot of the biodiversity that we have in our gut, and in many times it opens the way to colonization by pathogens. So once the gut has been cleared out, we can have these pathogens come in and colonize. Now, since I'm at Memorial Sloan Kettering, one of the big advantages of being in a setting like that is that we have a very large patient population. We get about 200 bone marrow transplant patients that come in for treatment. And these patients, they typically have leukemia or lymphoma. And the goal of the bone marrow transplant is to reboot the hematopoietic system. So these patients will receive very large doses of chemotherapy to basically turn off their system, and then they get a transplant of stem cells to try to reconstitute the system and hopefully get rid of the cancer. Now, while this process is going on, the patient is immunocompromised, and so receives a lot of doses of prophylactic antibiotics and also antibiotics in response to symptoms of infection. And the idea that my colleague, Eric Paymer, had 6 years ago, right before I joined Sloan Kettering, is whether we can leverage microbiome biology, and especially this field was really starting to pick up at the time, whether we could use metagenomic sequencing to understand how the microbiota of these patients is changing over time, how it changes as a consequence of antibiotics, and whether we could really determine how these changes affect the risk of infection by certain pathogens. Here I'm showing the case of one specific bone marrow transplant just to illustrate how this type of data looks like. So these bars here, these colorful bars, they're results from 16S amplicon sequencing. So they basically represent the taxonomy of the fecal microbiome, and the samples were taken at different days relative to the bone marrow transplant. So this sample here was taken 9 days before the transplant, then 1 day before the transplant, and so on. And so the different colors here in these bars, they represent different taxa of microbes. Here they are clustered loosely at the genus level, and the fact that this sample at 9 days before the transplant has many different colors, that represents that the gut microbiome is very biodiverse before the transplant, actually with a biodiversity level that is similar to a healthy individual. However, as we approach the day of the transplant, the doctors start prescribing different antibiotics. And so these are what is horizontal bars here represent their timelines of antibiotic treatment. And as you can see here, these patients are exposed to several antibiotics of different classes, and that has profound consequences on their gut microbiota composition. So the gut microbiota composition starts changing, and especially here, 6 days after the transplant, you see what we call a domination of the gut microbiome by a single microbe of the genus Enterococcus, so this green bar. And actually this Enterococcus was a vancomycin-resistant Enterococcus that not only dominated the gut, but then entered the bloodstream and caused a systemic infection, a bacteremia, which is represented by this bar here. And so what we see over and over with these patients is that antibiotic therapy many times has this adverse consequence of enabling expansion by certain microbial groups that then can enter the bloodstream and cause a bacteremia. In this particular case here, then the doctor stopped vancomycin and he saw the other groups starting to come back up. here at the end of this observation period, it seems like Enterococcus was starting to come back up because of some other reasons. Okay, so we started doing this in large scale, and the first study that we published a few years ago, we analyzed about 100 patients, and we see all sorts of problems occurring with these patients, where they receive many different antibiotics in strange combinations. We have examples of patients that somehow managed to maintain their biodiversity of the microbiome throughout most of the treatment. Here we have an interesting case of a patient that had very high biodiversity, and then here coinciding with the administration of metronidazole, we have an expansion of Enterococcus of the green bar. We have cases of Enterococcus domination. We have cases of Streptococcus domination. So all sorts of interesting domination events, many times preceding a clinical infection. Of course, at this time, the doctors, at the time of the transplant, the doctors did not have the microbiome information in real time. So this was all done a posteriori. But what we aim to do in the future is that as sequencing technologies advance and become more powerful and cheaper, we hope to be able to do this in quasi-real time and use it as an approach to assist in the design of rational antibiotic therapies. Now, in this first study, when we looked across all patients, and these are the microbiota from different stool samples, irrespective of the patient, we can cluster them according to how similar they look, and immediately some patterns start to come up. So we see certain samples that are enterococci dominated. We have some samples that are very biodiverse, samples that are dominated by different types of microbes. But what we see here, what this black bar represents down here, is that pre-transplant samples tend to be in this biodiverse cluster that has a lot of Plautia, this pink microorganism, but a lot of other microbes. And then after the transplant, the patients can end up in different domination states. Another way to summarize this is if we synchronize all the patients according to the transplant day, we aligned all their data according to the transplant day, relative to the transplant day, and we just plot a single data point which represents the biodiversity of that sample. We start seeing this trend that the biodiversity before the transplant is high, as you'd expect from a healthy individual, even though these are diseased individuals that are up for bone marrow transplant, and then starts decreasing because the patients start getting antibiotics even before the transplant. And then, on average, the biodiversity of the gut microbiome stays very low after the transplant, although there are some exceptions. Some patients manage to recover or never lose their biodiversity. So we developed computational methods to try to understand what factors explain why some patients lose their biodiversity while others don't. And we looked at many, many different factors. This is just to illustrate one of the many factors that doesn't play a role. So this is the source of the stem cell that's used for the transplant, whether it comes from umbilical cord or all other types of transplants. And here, it's a particular case where there's no difference. So either the patient received a bone marrow transplant from umbilical cord or from any other source, both cases, the biodiversity drops and there's no statistically significant difference. Where we did see major differences is in the antibiotics. And here we're separating according to whether the patient received bacteriolactam antibiotics anytime during the treatment, which are this red line here where the diversity drops, and the blue line are patients that never received any bacteriolactam during their entire treatment. We see a very significant difference. So across all the variables that we could get our hands on, we saw that the main factors affecting the diversity of the microbiota are really the antibiotics that these patients take. And I like to think of myself as a systems biologist. So I want to understand this problem from a systems perspective. And so we need to think about these three components, the input, the output, and the system itself. So the input in this particular problem are the clinical variables, what kind of events the doctors manipulate that the patients have to go through. And here we're specifically interested in the role of antibiotics. Then the output is, of course, whether the patient gets the disease or not. And what we can do now with metagenomic sequencing is open the window into this system, the black box, which is the composition of the gut microbiota. So we want to understand how changes in the gut microbiota composition explain how different patients exposed to different type of antibiotic therapy will get the disease or not. So to do this with ecological principles, we can adopt mathematical theory that ecologists developed. And here I have an example of dynamic equations that aim to describe a minimalistic version of the gut microbiota and the competitions that go on in this ecosystem. So the system is represented by 4 different equations. One of them describes the dynamics of the nutrients in the gut. And then we represent the bacteria that are in the gut according to their level of sensitivity to an antibiotic. So we classify the bacteria according to whether they're sensitive or tolerant to a single antibiotic. And then we also model the level of the antibiotic in the gut. What we get by using this minimalistic mathematical model is that the model predicts a concept that is also known from ecology, which is a concept of bistability, which means that there are different types of compositions of the gut microbiota that are stable. For example, we have this stable state here that is a domination of the gut by antibiotic-sensitive bacteria. And this, we imagine, is what happens to most healthy people. There's a diversity of microbes that are normally sensitive to most therapeutic antibiotics. And this is what ecologists would call a stable ecosystem state, because small perturbations around this state, our mathematical model predicts, would lead back to the same state. So, for example, if you take mild doses of antibiotic, you'll end up in this state again. However, the mathematical model also predicts that there will be an unstable state somewhere in the middle, and beyond this unstable state, the next stable state, the closest one in composition, will be domination by antibiotic-tolerant bacteria. And the problem here is once you cross this unstable point, you can get stuck in the nearest steady state, and it's difficult to get out of that. Okay, another metaphor to try to explain that. Think about this valley as these two valleys here representing the different stable fixed points of the ecosystem. Here we have a healthy microbiota that is dominated mostly by antibiotic-sensitive bacteria, our commensals. And then if you perturb this microbiota, for example, by subtherapeutic doses of antibiotics, you might not leave that system, or by changing in diet, you might not leave that system. But a strong perturbation might get you into an alternative stable point where now perturbations will not let you leave that system easily. So you might need a very strong change in the microbiota forced in order to be able to recover again the healthy microbiota. Okay, so this is all ecological theory. How can we test that experimentally? So here I started working with a MD PhD student who was supervised by Eric Paymer. His name is Charlie Buffy, and now he's back to medical school to finish his medical degree. But during his PhD research, he conducted this very simple perturbation experiment. So he took a colony of conventional mice, and he gave them a single dose of the antibiotic clindamycin. And then he followed these mice over time, taking one mouse out of the colony at each of these time points, sacrificing the mouse and looking at their gut microbiota composition, both in the ileum and the cecum. I'll focus mostly on the cecum because that's where most of the interesting effects happen. So this is a perturbation experiment where we perturb the gut microbiota and then we follow it over time and see what happens. Now, another experiment which concerns more colonization resistance that was done side by side was take the same type of conventional mice, give them a single dose of clindamycin, and then one day after give them spores of Clostridium difficile. And then another control experiment. Now we have mice who did not receive antibiotic, but they received spores of Clostridium difficile. And so when we compare these two sets of mice, the mice that both of them received Clostridium difficile, but one set was pretreated with antibiotics, we see a very strong difference in the survival of these mice. So the mice with their pretreated microbiota, antibiotic treated, 40% of these mice die within the first 5 days, whereas from the untreated arm of the experiment, all of these mice resisted Clostridium difficile infection, and all of them survived the study. Now, we analyzed the gut microbiota and how it changed over time. And here again, these are biodiversity timelines for the cecum of the mice. And here, the blue and the red lines, they represent the biodiversity. These points are before the mice received clindamycin. And then the mice received clindamycin, and the next time point at day 0, their biodiversity was very low and failed to recover pre-clindamycin levels, even up to 28 days after clindamycin. Now compare that with the untreated mice, who maintained their biodiversity very high throughout the study. Okay, and so this drop of biodiversity happened both for the mice that received Clostridium difficile and not. Both of them received clindamycin, but all of them had a very sharp drop of biodiversity. Okay, so now we're going to look into the actual bacteria that are in the gut, and we do this using metagenomic sequencing. We get a bunch of sequences that we can classify, and this somewhat busy slide represents the 3 arms of the experiment. So these are the mice that received Clostridium difficile, but they were not pretreated by clindamycin. So each of the rows here represents a different microbe, and each of the columns represents a different time point. So their biodiversity is very high. You see a lot of different microbes. The gray bars represent a missing microbe, a microbe that was not detected. And you see that there are a lot of different microbes, and they tend to stay stably the same microbiota composition throughout the whole study. Now this arm here represents the mice that received clindamycin only. And before clindamycin, their biodiversity is comparable to the mice that never received clindamycin. But then after the antibiotic, most of the microbes are missing, right? So you see gray bars. And interestingly, even up to 28 days after clindamycin, single dose of clindamycin, these microbes didn't return. Now, this here is the arm of the mice that were treated with clindamycin. Again, the same thing, most of the microbes vanish, but then they received spores of Clostridium difficile. And this row here represents Clostridium difficile, and you see Clostridium difficile coming up above detection. So it starts to colonize the gut and establish itself. And if you compare that with these mice here that also received the same dose of Clostridium difficile, but they had an intact microbiota, you can really see the difference. So these mice lost their ability to resist colonization. Okay. Now, the data from the previous slide was actually pooled data where each of these 3 arms had itself 3 replicate experiments. So they had each 3 cages where we had the same experiment going on side by side. And an interesting analysis that comes to look in detail at each of these 3 replicates is that the dynamics of the microbiome, the changes that occur after clindamycin, are actually very reproducible. So the first plot here represents the pooled data, so the average microbiome composition across 3 replicate populations. And here we're detailing the 3 replicate populations. And here, what looks like random changes that happen after a perturbation. So you have first a very biodiverse microbiota, then you give clindamycin, and you see domination by Enterobacteriaceae, then different bugs coming up. This is Akkermansia, this is Blauthia. So you see these bugs coming up and down, different successions, and this could just be random responses to a perturbation. But when you look at the 3 replicates down here, you see that all the different mouse colonies went through the same domination events, the same successions. And that really suggests that it's not random what we're seeing, but it's actually the deterministic result of ecological interactions happening between the bugs that colonize the gut microbiota. And so inspired by that, we decided to model the ecological dynamics in the gut microbiota. And the principle for this is actually quite simple, in spite of the scary looking equation. But the idea is that we want to describe how each of the microbial taxa is changing over time by modeling 3 different terms. Term number 1 is how much that microbe grows. That means if that microbe was alone in the gut, how fast it would be growing exponentially. So we want to determine the exponential growth rates of each of the bacteria. Then the second term here is the interactions between the microbes. And we include terms for pairwise interactions between every pair of microbes. And then this third and last term here is the effect of the antibiotics in every single microbe. So this type of mathematical model, is again borrowed from ecology, and it's called a Lotka-Volterra framework. So it's a very, very fundamental concept in ecology. But the idea here is that we take massive amounts of data, as many as we can get our hands on. We have these temporal dynamics, and then we use modern machine learning techniques to parameterize these models using methodologies that help to avoid overfitting. And that enables us to calculate from in vivo data the exponential growth rates of the bacteria, how the bacteria are interacting with each other, and the effects of the antibiotics on the bacteria, all from in vivo data. So this is actually real effects of how the antibiotic is affecting the microbial community. Now, this interaction matrix that is telling us the pairwise effects of how each bacteria is interacting with every other bacterium is already very informative because it gives us putative mechanisms of how bacteria keep an ecosystem stable. But once a model is fully parameterized, and if all the benchmarks for parameterization pass certain tests, we could eventually even use this to predict novel time series. So this means, for example, we could use it to assist doctors in deciding which antibiotics to prescribe in a clinical setting. So this plot here demonstrates that the model does have predictive power. So this is a result of a cross-validation technique. So it's a validation, a computational validation of the model, where we're comparing the actual data with the predictions of the model. And you see that there's some correlation. Here, the different colors of the dots represent how well the model can predict the microbiome a few days in the future. And the correlation is very strong if you want to predict 2 days in the future. But of course, if you want to predict up to 30 days in the future, you can still predict it with a certain predictive power, so a certain correlation, here 0.62. But of course, that prediction degrades over time. And this is pretty much like trying to predict the weather. Of course, your weather forecast for tomorrow is probably pretty accurate. But if you want to forecast the weather in 2 or 3 weeks, of course, it's going to be much worse. But this is showing that it is possible to try to predict the gut microbiota with computational models. And then when we look at the interaction matrix that we obtained from our computational modeling, we can look at how the bugs are connecting to each other. What are the signs of their interactions? And so this is a putative model that we could extract from our Lotka-Volterra model using the mouse experiments. So the model is predicting that there's mainly these 3 groups of commensals that interact in the gut microbiota of conventional mice, and that they somehow, through a mechanism that we cannot tell from the data, but there's a negative interaction sign that is telling us that Coprobacillus is keeping Enterococcus at a low level. Now, once we give clindamycin to the mice, what is happening is that it's affecting these 3 microbial groups and causing them to go down in abundance. And once these guys go down, this guy can come up and dominate the gut microbiota, and that seems to open the way to invasion by Clostridium difficile. Okay, so this is the putative model that comes from analyzing our Lotka-Volterra dynamics. Another outcome from the model is to allow us to predict what would be the next stable state of the gut microbiota. This is a somewhat abstract concept because there's no such thing in the end as a really stable state of the microbiota. This is what would happen theoretically if the mouse was left alone. And it's telling us that if left alone without any antibiotics, this would be the composition of its gut microbiota. This would be the composition after perturbation by clindamycin. So the model is predicting that the mouse will not recover the original composition. And this is the composition that the model predicts the gut microbiota would converge to after it has been exposed to clindamycin and then Clostridium difficile. So it's predicting that Clostridium difficile would actually increase into abundance enough that then would co-colonize the gut microbiota and reside with the rest of the microbiota there. This is, of course, just a modeling prediction. But then what we did next is we scaled up our mouse experiment, whereas originally we carried it out only with clindamycin. Next, we took a much larger number of mice and we give them different types of antibiotic treatments to generate diversity. And we also waited different times until we gave them Clostridium difficile spores. So we did this large scale, or rather Charlie Buffy did this large scale experiment, and then we took all these mice together. We looked at how much Clostridium difficile managed to colonize their gut by measuring Clostridium difficile CFUs in the stool, and then we looked at the microbes that were in the gut, and we tried to correlate the levels of commensals with the ability of Clostridium difficile to invade. And this is a list of the microbes that passed statistical significance. At the top of this list is a bacterium of the same genus as Clostridium difficile, Clostridium scindens. It has a very strong anti-correlation with the ability of Clostridium difficile to invade. And the red bar here actually represents a detail that's quite important, which is that we found that correlation holds up both in cases where the biodiversity was high, but even in certain mice where we saw that the biodiversity was low, but they still had Clostridium scindens, and those mice still managed to have a very high resistance to colonization. So this was telling us that Clostridium scindens is indeed quite important in the colonization resistance. What we did next is we went back to our bone marrow transplant population, and we selected a subset of the patients for which we had evidence that there was exposure to Clostridium difficile by using a more sensitive PCR test. So these were all patients that we were sure were exposed to Clostridium difficile, but not all of them developed Clostridium difficile infection, clinical Clostridium difficile infection. So then we could ask the question, what is the difference between these patients? Is there something in the rest of their microbiota that explains why, in spite of the fact that they were all exposed, some of them got sick and others didn't? And so we did a large-scale Lotka-Volterra modeling where we took all the commensals that were present both in mice and in patients, and we built models that we could compare, ecological network models that we could compare with each other. And then we selected the bugs from within this model for which we had evidence both from the mouse model and from the patient population that they protect against Clostridium difficile. And again, Clostridium scindens came on top of that list as one of the bugs that conferred most of the protection. So then Charlie Buffy decided to explore this further with targeted mouse experiments where he treated the mice with antibiotics, and then he colonized the mice with Clostridium difficile. But in some of these mice, he tried to reconstitute their microbiota. Some of these mice here were control mice where rather than reconstituting, he just gave them a dose of sterile PBS. And so then Clostridium difficile can colonize this gut very well. And these are mice where he gave a cocktail of 4 bacteria, including Clostridium scindens, so Clostridium scindens plus 3 others that were at the top of our list as protective microbes. And these microbes seem to keep Clostridium difficile away. And these are mice that receive Clostridium scindens alone. So Clostridium scindens alone protects also the mice quite a bit. Not as much as the cocktail of 4 bacteria, but it keeps a lot of the protection. And these are, again, survival curves, so showing the mice that receive PBS alone, pretty much like in the case for clindamycin that I showed before. A lot of these mice die within the first 5 days. These are the mice that got the cocktail of 4 bacteria, and all of them survived the invasion by Clostridium difficile. And these are the mice that receive Clostridium scindens alone. So some of these mice do die, but there's a strong boost in the resistance to colonization and against infection by Clostridium difficile. So this is a very interesting finding, I think, in many ways. One of them is that it gives us hope that we can single out particular microbial species that protect against Clostridium difficile infection. But another very important observation is that at least this microbe here doesn't explain 100% of the protection, and the cocktail, including that microbe plus 3 others, protects even better, which tells us that colonization resistance is very much a community feature, right? That we require certain ecological interactions that we don't fully understand yet to convey resistance to colonization. And so that's very, very interesting, and I think opens up a lot of interesting questions for the future of microbiome biology. Now, once you do single out a certain microbe, you can try to drill down even to the level of molecular mechanism. And Charlie Buffy continued his experiments eventually to determine that the secondary bile acid metabolism was a key feature of Clostridium scindens that seemed to be very, very important to keep Clostridium difficile from invading, and especially the one coded in this bi-operon. He also did some interesting ex vivo experiments where he used cholestyramine columns to extract primary bile acids. And so in these bacteria that received Clostridium scindens that were exposed to cholestyramine treated stool samples, these were not capable of producing secondary bile acids, and so their ability to resist colonization by Clostridium difficile was also depleted, was also gone. So really singling out that the molecular mechanism by which Clostridium scindens prevents Clostridium difficile is through secondary bile acid metabolism. So I think this is a type of work that I hope would make Koch proud, in the sense that we can go and isolate single sources of single microbes that explain a lot of the phenotype, but at the same time reveals to some extent the limitations of Koch's postulates, in the sense that it shows that it does not explain fully 100% of the phenotype. So as we move on, what I hope that we can do over the next 5 to 10 years is to really expand these type of approaches with many more data sets to start to see the signal within the huge noise that comes from patient type of population. Again, here we can leverage the fact that at Sloan Kettering we have about 200 bone marrow transplant per year, and the fact that this patient population is particularly fragilized in the sense that they have very high infection rate. So it's about 1/4 of these patients get infections, bloodstream infections. So it's by following a few hundred patients, I mean, a few hundred is still a lot, but by following a few hundred patients over time, we can get a lot of cases and really ask questions about how variation in the gut microbiome explains a risk of infection. This, of course, will be leveraged by the powerful technological improvements in next generation sequencing. This is a slide that I imagine many of you have seen, or versions of it, that is comparing the drop in sequencing price, comparing that with the price drop that comes from Moore's law, the well-known Moore's law that explains how computers and iPhones and all that get cheaper and cheaper every year. And to help us really think about how something that's advancing even faster than Moore's law can bring really disruptive technologies, let's just think about how Moore's law has changed our lives. So if you look at the type of calculations that were done in the 60s, you see, my father, when he studied engineering in engineering school, his major calculation tool was this calculation ruler. And computers were restricted to certain departments of electrical engineering in certain universities, and they were mainly mainframes that occupied whole rooms. Now, when I studied engineering, this was my calculation tool, already much more advanced, probably more advanced than some computers in the 60s. And now these days, most students will have something like this, and a lot of us will have very powerful computers in our pockets. Now, think about how this level of technological advancement that has changed our life, and now we take it for granted, how that can be applied to thinking about sequencing, and so how even faster advancements in sequencing technologies can enable new technologies in the years to come. Right, so this is of course just a caricature of what we can think, but within 5 to 10 years, technology will be cheap enough that we can apply it in a bedside manner and just follow the gut microbiome of patients. I mean, a toilet like this, of course, will be very expensive, but I just bought a machine like this to my laboratory. It costs $85,000, so it's not that much if you take into account the budget of a laboratory. And my wife, she's an architect in New York City, and she had clients that are very rich and live in places like the Trump Towers, and they would shell out $85,000 for a toilet if that bring them better health, no problem. So now imagine what we can do with this type of technology if it advances fast enough. Now, as our study progressed, this is at Sloan Kettering right now. So as our study progressed, when we first published the results that I showed you before, we had about 100 patients. Since the time of that study, we continued collecting stool samples for these patients, and now we have patient timelines for about 800 patients like this. And so patterns that when we did analysis 4 years ago looked kind of fuzzy, like here the drop in biodiversity over time compared to the day of the transplant, will now become much, much more obvious. And we'll start seeing nuanced effects that were, before that, they were evasive, and now we'll start seeing those. Another example here are the number of positive cultures in blood samples from these bone marrow transplants. So basically signaling infections. And here there's a peak at about day 5 after the transplant. And this is the data from 2011, and now the data that we have right now is showing much clearer trends, including other peaks that we're very interested in finding out where these come from. Now, to help us do that, we have support from the NIH. So Eric Paymer and I are co-PIs on a U01 grant that we just got awarded. And here the goal is to increase our sequencing power even more and really dissect the systems biology of how the gut microbiota helps us prevent infections, mostly using the bone marrow transplants as our model. And another technology that we're bringing into play that I'm very excited about is metabolomics. And we're applying that to stool samples, and that enables us to find not only metabolite signatures of the microbes and how they're interacting with each other, but also signatures, for example, of the drugs that these patients are taking and how they're broken down by the microbiome. Here I have some examples here. I'm showing peak areas for certain antibiotics that the patients took. We knew that they took these patients and they took these antibiotics, and this is what these bars here represent. But the timelines here, they represent actual measured levels of the antibiotic in the stool. So that gives us a different level of precision to be able to develop new powerful models of how drugs are affecting the microbiome and vice versa, how the microbiome is affecting the breaking down of drugs and how they affect the outcome of patients. We're also doing fecal matter transplants, and we have right now a clinical trial going on. It's still early days. This is led by my colleague Ing Thor, and we have already 10 patients that went through the whole process. This involves, of course, a randomization process. So 5 of them received the fecal matter transplant, where 5 of them did not. And the idea here is that we follow bone marrow transplants, but as soon as they come into the clinic, we take a stool sample. And then after engraftment, we use that stool sample to try to reconstitute the gut microbiota diversity. And so these are the patients that so far we've got. These are patients that were randomized out, so they did not receive the transplant. These are the patients that received a fecal matter transplant. And this might be anecdotal at this point. The data is still very limited. But at least in these 10 patients that we followed, the ones that received a fecal matter transplant had a better outcome. So again, the statistics is low, but it's showing that at least we're not harming the patients by doing this. And here are some very early results. So these are timelines of 2 of the patients that were randomized out. So they had all the checks. They were up for fecal matter transplant, but then the randomization procedure decided that they're not going to receive the transplant. And so this is their natural microbiota progression. So you see their biodiversity stays relatively low. And these are examples of 3 patients for which we have already the microbiome data that did receive the fecal matter transplant. So you see this sample here went to reconstitute the gut microbiota here at day 50 in this case. And you see definitely some signatures of the early microbiota, and the same thing with these 2 other patients. So we have, evidence beginning to reveal itself that these fecal matter transplants do manage to work in these patients. So we look forward to this type of studies, as I think they're going to be very, very promising. So thank you very much for your attention. This is my lab at Memorial Sloan Kettering right now. As I mentioned, we work in different projects, including Pseudomonas aeruginosa social microbiology, and also cancer biology, and particularly Jonas Schutter and Catherine Coit, who were former PhDs with Kevin Foster. They just joined the lab in the past 3 months, and they're going to help us unravel the ecology of the gut microbiota. Thank you very much.